In this video, we're going to talk about the cell cycle. Now, the cell cycle just refers to a series of changes that a cell can undergo from the time that it's formed until it reproduces. And we can describe the cell cycle as being divided into two subphases, interphase and cell division, or M phase, or mitotic phase. Interphase is actually a period of time that the cell would typically spend the most time within, and it's basically where the cell grows and carries on its usual activities. Cell division, or mitotic phase, is where the cell actually divides in two, and we'll talk about the events that occur in each of these phases of the cell cycle. So this just shows the cycle. Remember, as a cycle, it's a circle, and you just got to pick a starting point, right? So for the sake of this description, we'll say that the starting point here, right after the end of cell division and the beginning of interphase. So you can see that all of this is interphase. And with this cell cycle, you can see that this particular cell is spending most of its time in interphase. And the first phase in interphase is actually G1, which is growth. So immediately following the cell division here, we find is that cells are able to grow and basically help to replicate more of the organelles and essentially just create a lot of the components that may need to have been replicated following cell division. Now, G1 is a restriction point, and this is where cells are typically held at until they're basically going to start to prepare for division. So G1 is essentially just a kind of a place where cells would hold at and while they're undergoing their just normal activities. And if a cell is prepared to divide, it essentially can continue on into S phase. So S phase is basically synthesis phase, where we have growth of cellular components, and DNA synthesis, where you start to replicate your chromosomes. S phase is followed by G2, and G2 is basically we have growth and final preparations for division until you get to this G2M checkpoint. And this checkpoint is basically another restriction point, but essentially a point where the cell can just make sure that all of its chromosomes are replicated, all of its organelles are replicated, and it's ready to divide. Once it passes this checkpoint, it's able to continue on to M phase, where there's a series of subphases of mitosis or cell division that then lead back to cytokinesis, where the cell divides, and then the beginning of interphase again. And this relates to cell theory because each cell came from a previous cell, right? So what we find then is that maybe this particular cell gives rise to a new cell, and then this new cell can actually just again go through interphase and then potentially also divide itself. Now, M phase is also called mitosis, and it's the division of the nucleus. That's what we talk about with M phase, is that although it's cell division, we're really going to focus on nuclear division during this phase, and it's where the DNA is actually duplicated and distributed to new daughter cells when the cell divides. And we can divide M phase into four major sub-phases. We have prophase, metaphase, anaphase, and telophase, or PMAT. And these are just specific phases of division that we can identify under the microscope. So let's start with early prophase. So this is the first phase of mitosis. And we can see that early on in prophase, our nuclear envelope is still intact. Our chromatin is condensed now into chromosomes that are somewhat visible under the microscope. So if you look over here, here's our nucleus, and you can see that there's actually visible chromosomes now because the chromatins start to condense. Our mitotic spindles start to form, where the centrioles actually have separated, and we have duplicated centrioles, and they form what's called an aster meaning star. And if you look at it, it kind of has this sort of star pattern of microtubules that project away. We call this the mitotic spindle because these microtubules play a role with anchoring to chromosomes and then helping to pull those chromosomes to opposite sides of the cell. Now, later on in prophase, what happens is the nuclear envelope degrades. And once that nuclear envelope degrades, the chromosomes basically spill out into the cytoplasm, and they're shuttled by microtubules onto this whole aster network of microtubules. We also see that the spindles form poles where they move to opposite ends of the cell, and this starts to prepare the cell for division in the sense that this side can pull chromosomes this way and this side can pull chromosomes this way. And We call the place where chromosomes bind to the microtubules the kinetochore. Kineto meaning movement, core meaning center. So the kinetochore is basically, a place where these chromosomes can essentially bind to microtubules and then move in a particular direction. Now the next phase of M phase is metaphase. Metaphase is identifiable because our chromosomes line up at the equatorial line, or sort of the midpoint of the cell, and they form what's called the mitotic line here, a metaphase plate. So the metaphase plate is essentially where all your chromosomes line up in a particular line here, or row, and that the microtubules on either side can actually start to pull on either end of this kinetochore. And what we'll find is that each copy of the chromosome will eventually get pulled to opposite ends of the cell. So that's what we see in anaphase. So during anaphase, the chromosomes get pulled towards opposite poles here, where this daughter chromosome gets pulled to this side, this daughter chromosome gets pulled to this side. So what happens is you get a complete set of chromosomes being pulled to either end of what will become a new cell. And that way, this eventual cell will have a complete set of chromosomes, and this eventual set will also have a complete set of chromosomes. And this is actually anaphase, where they begin to get pulled apart towards opposite ends of the cell. Now, the last phase of M phase is called telophase. And telophase is characterized by the formation of what we call a cleavage furrow. And this cleavage furrow is basically where the cell starts to kind of form an infolding here, which will eventually kind of pinch away and form two cells. Also during telophase, the nuclear envelope is reformed. That way chromosomes are repackaged into a new nucleus. And this contractile ring starts to occur at the cleavage furrow as well. And then the last part of cell division is cytokinesis. Cytokinesis is not technically part of M phase, but it's necessary for the cells to divide. Cyto meaning cell, kinesis meaning movement. So cytokinesis is basically where the two cells kind of pinch apart. And then what you're going to have is basically completely opposite cells there. So cell division is controlled by different signals. And we need go and stop signals that direct when a cell should or should not divide. Go signals include things like surface to volume ratio of a cell. So if cells become too large, where there's an inadequate amount of membrane for exchange, this can be a trigger for cells to divide. You're going to see this in things like adipocytes or fat cells. So when fat cells become large, that itself can trigger the formation of more fat cells, which means they can store more fats. Other chemicals can also trigger cell division. Things like growth factors or hormones can stimulate cells to divide. And otherwise, we need stop signals because of the 100 trillion cells in our body, we don't want all of those dividing at once. And so some of the stop signals include things like the availability of space in a tissue. If there's not a whole lot of space, then cells stop dividing. Otherwise, normal cells can stop dividing when they come into contact with other cells, and we refer this as contact inhibition. And that way these cells kind of understand at the cellular level that there's no more space for these to divide, and they've occupied all the space in the tissue. We need checkpoints for these key events, and these checkpoints are basically things that allow cell division and the cell cycle to occur appropriately. So this G1 checkpoint we call restriction point. It's the most important because it actually is basically what prevents the cell from continuing to divide just unchecked. And if the cell does not pass, then it enters what we call G0, which is basically where no further division occurs. There are lots of cells in your body that are currently in G0, especially neurons, because neurons don't really divide for the most part, brain cells. And so most of your brain cells are in G0, which means that they're still in the cell cycle, but they're not preparing for division, and they're just kind of undergo their normal activities. So just looking at these different checkpoints here, remember G1 is this restriction point. This is a place where cells are held, and then there's further processing to make sure that cells either shouldn't divide, and if they don't, then they enter G0. If they can divide, then they're allowed to continue on to S phase, where you can replicate your DNA and grow more organelles. Then you end up entering G2, where your cell can actually start to grow additionally and also prepare for division by making sure that all DNA has been replicated and all your chromosomes have been replicated. And this G2M checkpoint is just a nice last checkpoint to make sure that everything is ready to go for cell division, and then we re-enter M phase. Remember, M phase is mitosis, where we have prophase, metaphase, anaphase, and telophase, followed by cytokinesis, where you have the cells actually splitting into two separate daughter cells.